Chapter 31, Section 2, Streptococcal Diseases. We're going to start off here with this section with one of my favorite bacteria. Let's talk about Streptococcus pyogenes. Now, what they did is they took the different species and subspecies and strains within the genus Streptococci, and they grouped them more like with like. Pathogenic capabilities, how virulent are they, how much damage do they do, who has what abilities, and they grouped them all together. Turns out Group A, like we have right here, Group A are the big bads, the most virulent, the most damaging, has the most capabilities and whatnot. These are going to form wounds that are going to be pus formers. Hopefully you remember from biology at some point in time that pus is not a bad thing. People look at pus and go, oh my God, it's all pus. Okay. If it's coming out white pus and it doesn't really have a bad odor, that means the majority of the pus is your white blood cells and plasma from your blood coming out. And that fluid is antibodies, complement proteins, clotting factors, and whatnot. You know, it's full of white blood cells, macrophages, monocytes, I know, tomato, neutrophils, natural killer cells, and all sorts of other blood cells. Well, it's when it starts to get colored or starts to have really bad smells to it that now it's more bacterial and you get these overgrowths and all sorts of stuff. We'll talk more about that later. The name, Streptococcus. Strepto, Greek for chain, and that is literally how these things grow. As you see here in this computer-colored electron micrograph, that you see the beginning of the cellular division. The cell wall here, the peptidoglycan layer, is dividing along the equator of the cell, and it does so in a pre-planned axis. Okay, so there's always this plane of division, which will allow the cells, daughter cells, after the post-division daughter cells, to be in line with the rest of the chain. This chain breaks in half, no big deal. The two parts will begin to grow, grow the population and continue making more longer chains. One of these terminal bacteria here fall off, not a problem. It will divide due to, pre-planned based on cytoskeletal proteins. Here's the plane of division. Now the two daughter cells, those will divide, and they become four, and they will all divide so that their planes of division are parallel to each other, or as close to parallel as possible. So you get this long chain growing. And you can see this chain on gram-stained slides. If you were to be careful in the preparation of your bacterial smear and your heat fixing, you can maintain the bacterial cells being such close proximity they remain attached to each other, and you can actually see the streptoness. The pyogenes, the species, is based on where they were identified, how they were identified, what they were taking from, whose name, and all sorts of stuff like that. The pus. This wound you see here, this gaping wound that is leaking discolored pus. And you can see darkened tissue in there. That's not, oh, look, those are veins. No, that is dead, decaying tissues. As tissues in all mammals die, it turns black. Blood flows in, blood doesn't flow out. You get the buildup of waste products. The cells around that area begin to die. They go necrotized. They die, they burst, causes what red blood cells there to also burst. You get the iron from the heme turning discoloration, clotting, and all sorts of stuff. That whole domino effect, the things that I just talked about, can all be triggered by the lysis of red blood cells. Hey, guess who is very beta hemolytic? Remember beta hemolytic? That's kind of what we talked about with the blood agar plates, alpha, beta, gamma. kind of, sort of hemolytic, total hemolytic. No, hemolysis, alpha, beta, gamma. So this beta hemolytic streptococci means they are going to lyse red blood cells that from the blood that flows in, starts to lyse, triggers clotting factors, you get the clot, so blood flows in, nothing flows out. Build up the waste products, cells start to die, tissues die, they become more liquidy, and then the bacteria has all this nice soup to grow into. And a lot of times students will ask, well, what about oxygen? This is going to be really gross and anaerobic. Yeah, remember, Streptococcus are one of those that, oxygen's there, okay, but they'll do very well without it. Talked about Streptococcus pyogenes once before in the lab when I gave you the stories about the guy who lost his arm because of necrotizing fasciitis and said that the Streptococcus pyogenes, they isolated from the wound when they went through and did the different amputations of his arm. They traced that to his throat. Yeah, Streptococcus pyogenes is normal flora of our throats. It is an opportunistic pathogen that has been shown to cause strep throat. Strep throat is different than, oh, I have a sore throat, because sore throat is a symptom of strep, and as we're going to see on the next slide, strep throat also has lots of inflammation as well as some minor tissue damage due to some of the enzymes and toxins released by Streptococcus pyogenes. We also see that, and forget the throat, let's talk the skin. If you have Streptococcus pyogenes on the skin, and you'll see me, I will keep referring to the skin, and I'll be touching my face, one, because that's this little camera, that's what you can see. But the other, we see impetigo and erysipelas more often than not associated with the face. Why? Well, guess what? I'm touching. I'm coughing on my hands. I'm touching my face. So whatever was in my throat, it's now in my hands. It's now in my face. I am itching my beard, scratching myself. Little kids, adults, all do this. Impetigo, you're going to see the bacterial infection. The bacteria are growing in the epidermis. Their presence, their exoenzymes, the enzymes they're producing and excreting out, are damaging the epidermis. It's going to cause a small raised rash area that gets very weepy, gets very scabby. If they get down below the epidermis, down in that between the epidermis and dermal layers, and they start spreading, you get to see what is referred to as erysipelas. Erysipelas, and you're not going to see the scabbing like you would with impetigo. What you're going to see here is a large area that is now red and inflamed, extremely hot to the touch. We'll see that in a few minutes. The strep throat. All of these little pixelated red spots you see right here. This is areas where the capillary beds are bursting. You go back there and you may actually, shine a light even further back and you look there, you may be able to see areas where the mucous membrane here is disrupted because the bacteria that is growing on the surface is doing damage to the tissues. Really damage to the tissues. All of these areas of capillary beds bursting, you're going to have a release of bradykinin and others that are triggering an inflammatory response, and also our tissues and our brain recognize that bradykinin release as pain signal. Part of the reason why it hurts to swallow when you have strep throat. As you saw in lab, well, you don't want to sit there and swab the test right around here because at the tonsils and the uvula, because this is also coated with all the bacteria of the mouth, and any Streptococcus pyogenes present there is just going to be overrun by oral. So if you try to swab there and run the test on it, you're going to get a false negative. So what you need to do is swab back in there. Yes, for that, truly to do that correctly, you're going to need a tongue depressor to press the tongue out of the way, because again, you want nasopharynx bacteria, not oral bacteria. And you need to get back in there with that swab, touch the back of the throat, the nasopharynx, and that is all. When you look at the impetigo, as I said, small raised area looks like a small rasp pustule. This is more epidermis, almost down to the dermal layers. Could have started off with a scratch right there, and as the bacteria spread in different places, because it's spreading under, the epidermal layers, spreading because it's itchy and you scratch and you was here and you now moved it down there. This will clear up, minimal chance of scarring. If it doesn't clear up quickly, it can continue to spread, and the longer it remains there, increased chance of pockmarks, scar tissue, and things like that forming. And this here is the impetigo. This will literally cover half the face, if not the full side of the face, and the nose. What you have here is this started off as a small area someplace right here on the cheek maybe, which could have went impetigo, but it actually went deeper down to the dermal layers, and in those layers between the epidermis, dermis, the underlying adipose tissues, none of these are actually linked together. They're just laying on top of each other. They're held together by fibrous tissues, but they're still distinct from each other. So you can get the spread of bacteria between these layers very quickly. And what's spreading even faster than the bacteria in these layers are the toxins and the enzymes produced by these bacteria. These will diffuse at a faster rate. So you get those spreading, and then ahead of those, you're going to get the inflammatory response that's spreading even quicker. Red, raised, it's swollen and it's painful, and you go to touch it, it is very, very hot. That is our immune system there, kicking up the thermostat of the inflammation, trying to literally cook, burn out, denature all the bacterial proteins and vegetative cells there. Now, one of the things we find with bacterial infections, especially ones that are as highly pathogenic as Streptococcus pyogenes, Staphylococcus aureus, which we'll talk about in a later section of this chapter. The bacteria get in, cause an infection, which is damaging in and of itself. But the problem is, once they're in there, they start to produce bacterial toxins, bacterial exoenzymes and things, and even just chunks of the bacteria itself coming off, spread. They get into the bloodstream, you get toxemia. Okay, what's toxemia? Rule of thumb, anything that ends in emia means blood. So blank emia means whatever blank is, it's now in the blood and is spread throughout the body. Streptococcus pyogenes, we see what is referred to as streptococcal toxic shock syndrome. This toxic shock syndrome. The bacterial infection was localized, but the toxins, the exoenzymes, went systemic all throughout the body and are causing damages everywhere. Part of that, our body's response to that extreme damage everywhere, is to go into shock. You got to remember, shock is deadly. Means your blood pressure skyrocketing as your blood volume is decreasing because you have head to toe inflammation. All the fluid volume is leaving your bloodstream and entering into the tissues. So your heart's beating faster trying to compensate, and your brain's telling other parts of your body to shut down to keep whatever blood is still there going to your brain, your heart, your liver. This continues on long enough. After a few minutes, you start to see this cascade effect of organ failure. Usually starts kidneys, then the liver, then heart, brain, dead. And that's all because you had a mild bacterial infection. We saw this streptococcal toxic shock and some other toxic shock syndromes were tied to the use of tampons back in the 80s, early 90s. Turns out that the bacteria, the tampons had bacteria on them. Inserted into the vagina, the bacteria would start to overgrow the normal flora there. You have lots of open wounds as women are shedding the layers, the linings of their cervix. and their uterus, I'm sorry, not the cervix, shedding the lining of their uterus. Bacteria get in, that stays relatively localized, but the toxins went systemic. I actually had quite a few young ladies die from this. Could also, because of this toxic shock syndrome, and a milder version of, not quite as deadly, you get this, what is called scarlet fever. Scarlet fever. You see here in this young boy, the face looks flushed. It's a permanent flush. As long as they're diseased, as long as they have scarlet fever, their face will remain flush. It looks literally like they're embarrassed. They're just blushing, but it's a very dark, maintained blush, not for the few minutes like, oh my God, I just embarrassed you. You look at you blush. This is a long drawn out. And if you look, this blush actually goes down the neck to the torso, down the arms. You go past the face, which is highly vascularized, so it looks more like a blush there. You look here down the nose, down the neck, onto the torso, the chest. This blush here, this reddening here, is actually thousands and thousands of little spots. Spots accumulate high enough, it gives them this overall red appearance. This is scarlet fever. Scarlet fever has a relatively low mortality rate. It's just has a high degree of the children who get it are incapacitated, which means mommy and daddy, one of them have to stay home to take care of them. And it's a very long, protracted fever, this scarlet fever, this disease, these symptoms, because it's going to take a long time for the body to remove all the toxins, the bacterial toxins, the bacterial exoenzymes and things like that that were produced and pumped out. With Streptococcus pyogenes, we also have to take into account that there's going to be some tissue damage. That is, there's always this potential. Depending on how deep into the skin Streptococcus gets, you could have, surface, like the impetigo. You can get down below the epidermis, the dermis, down between the dermal and the adipose tissues. If it gets that deep, you start to see what is referred to as cellulitis. And again, this cellulitis can spread rather quickly as it gets down below the adipose tissues and things like that. Because remember, as I said, it's just layers that are loosely held together. They're not intertwined. They're just discrete layers. So between those layers, the bacteria can spread. If it gets down deep enough, you actually begin to see tissue degradation, tissue death, the necrotizing. You get what's referred to as necrotizing fasciitis, the flesh-eating disease. This wound you see here could have started as a small cut, and it is now spreading down his leg. Now you see these nice demarked lines here. Chances are they went through and removed with a knife or a scalpel the discolored, you see right there, black tissues. Now, I'm not saying black because this person is African. I'm saying black because what happens is as the tissues die, the tissues, cells burst, cells cause what blood may still be there to clot. The red blood cells burst, the heme in them oxidizes, starts to discolor due to iron rusting and all of that. And the tissues and the clotting start to turn black. Black tissues are dead tissues. One of the things also, black tissues will cause tissues and cells neighboring that dead stuff to also die. So you'll see the beginning of the spread of the black tissue, the death of the tissues as it's spreading down the leg, and it is spreading up towards the abdomen, which is why you see wound here, wound there. Don't know which one came first, the wound here, the wound there, but it spread from one to the other, or vice versa. What's going to happen is you can see the discoloration of the tissues here, here, and here. This is going to continue to die and spread until they can get ahead of it. Now, since it's up here at the hip, chances are this is going to be mortal because you have lots of vascularization here. it's going to spread. Now, if this has been down, say at his ankle, they could have lopped off at the knee, maybe saved. I have no idea what happened to this individual. They don't mention in the textbook, but this is a very deadly infection, or the potential to be a very deadly infection. We also have, beyond Streptococcus pyogenes, we have Streptococcus pneumonia. Strep pneumonia, you'll hear it referred to that. It's just strep instead of Streptococcus pneumonia. They'll refer to it as strep pneumonia, or is one of the causative agents, probably the top 5 causative agents of what we call pneumonia. It's encapsulated, it has a capsule. Strep pneumonia is the one that you use by Griffin to do the Mickey Mouse test. Remember the ones where he had the R and the S strains injected in the mice? R, mouse lived, S, mouse died. Remember? Try to prove the transfer of something from S to R and gave us the idea of transformation. Yeah, here's the bacteria for that. Streptococcus is going to be identified by culture. Going to go through and culture. You're going to see something that is Streptococcus, cocci in chains, beta hemolytic. Because honestly, what you really just need to know is a group A strep. Turns out most of the bacterial antibiotics that work on Streptococcus pyogenes are going to work at strep pneumonia. Streptococcus pyogenes isn't going to cause a deep pneumonia in the lungs, just as pneumonia is not going to cause a skin infection. So they really need to know, is it group A, group B, whatever, and then they test for drug resistance. Will antibiotic A still work, or is the group A strep that they've identified here as the potential causative agent, is it resistant to this one? Then do we need to move on to B? And they'll test the different A, B, antibiotic A, B, C, D, E to see which one still has the greatest potential for killing the microbe. You see here is a stain showing, this blank area you see around the cocci. This is due to that capsule. This is the layer, that protective layer here, that keeps these cells hydrated, protects them against loss of water, but it also keeps antibiotics as well as, your immune cells away. And just once again, to kind of give you an idea, see this halo here. This is a red blood. This is a blood agar plate. All the red blood cells that should be here are now lysed around this colony. Nice beta hemolytic. They call this beta hemolysis.